Today, we are diving into one of the biggest questions in all of biology. How can our cells, which all have the exact same DNA, end up being so incredibly different?
I mean, really think about that for a second. Your brain cells and your skin cells. They have the exact same instruction manual, the same DNA. So how does that even work? It's kind of like having the same giant encyclopedia in every room of your house, but you only use the plumbing chapter in the bathroom and the cooking chapter in the kitchen. The blueprint is identical, but what you build is totally different. That, right there, is the puzzle of gene expression.
Well, the answer, it turns out, is all about control. And I mean serious control. Cells have this incredibly sophisticated system for regulating which genes are active and which are silenced. And this isn't just one checkpoint. It happens at every single step of the process, from reading the original DNA blueprint to making a copy, all the way to building the final protein. It's like a multi-layered security system that's constantly deciding what gets a green light and what gets a hard stop.
Okay, so how does this all actually work? Here's what we're going to cover. We'll start with the library of life, then look at some simple bacterial on-off switches. After that, we'll dive into our own more complex code, see how cells can intercept the message, and finally wrap up with why control is everything. Let's get started. All right, first section, the library of life. So why do cells even need to bother regulating their genes? Well, think about it. A cell doesn't need to be doing everything all at once, right? A muscle cell has no business making digestive enzymes, just like a skin cell isn't supposed to be sending nerve signals. Gene regulation is really all about efficiency and specialization. It's about using only the chapters you need from that giant DNA library to build the exact right cell for the job. Okay, let's get into our first real-world example, bacterial on-off switches. This is a great place to start because it's so, well, elegant. We're going to look at a classic story of survival, all about how E. coli bacteria decide what to have for dinner. So, bacteria are masters of efficiency. They're all about saving energy, and one of their cleverest tricks is to bundle genes that all work together into these packages called operons. The best way to think about it is like a single light switch that controls all the lights in a room. Instead of turning each light on one by one, you just flip one switch. The whole package gets turned on or off all at once.
Okay, so let's check out the famous lactoperon. These are the genes for digesting lactose, milk sugar. Now, by default, this whole system is switched off. There's a special repressor protein that's literally sitting on the DNA, physically blocking the machinery. It's a roadblock, plain and simple. As long as it's there, the cell can't read these genes. But what happens if the bacterium's main food source, glucose, runs out, and suddenly there's lactose everywhere? Well, this is the cool part. The lactose molecule itself acts like a key. It grabs onto that repressor protein, changes its shape, and poof, the repressor falls right off the DNA. The roadblock is gone, the path is clear, and now the cell can finally start making the enzymes it needs to digest this new sugar.
But wait, it gets even smarter. The cell's logic has a second layer. See, it's not enough to just have lactose around. The cell wants to be absolutely sure it's the best option. So there's a second check. Only when its favorite food, glucose, is running low does another protein, an activator called CAP, come in and give the final green light. This tells the machinery to go full steam ahead. So you need two conditions, lactose present and glucose absent. It's a beautiful piece of logic. So that's the bacterial system in a nutshell, a really simple, elegant on-off switch. But what about us? What about our cells? Well, if bacteria have a light switch, our cells have something more like a full-blown mission control panel with tons of dials, buttons, and layers upon layers of security. So let's just quickly compare the two. Bacteria are all about speed and efficiency. Their DNA is basically out in the open, ready to go, and they use those simple operon packages. Our cells, we have a totally different challenge. Our DNA is meticulously packed away, so just getting to it is the first hurdle. And activating even a single gene often requires a whole committee of proteins to agree, some of them acting like remote controls from way, way down the DNA strand.
Okay, so the very first level of control in our cells is just physical access. Our DNA isn't floating around like a loose string. It's wound up super tightly around these proteins, forming a compact structure called chromatin. When it's bundled up like that, it's basically locked down. So before a gene can even be considered for being turned on, the cell has to physically unpack that specific section of the chromosome. And that right there is a type of epigenetic control. It's controlling the genes without ever changing the actual DNA code. Now, once a region of DNA is unpacked and accessible, things get even crazier. We have what are called enhancer sequences.
These are little stretches of DNA that can be thousands and thousands of letters away from the gene they control. So how do they work? Well, proteins bind to these distant enhancers, and then the DNA strand itself has to physically bend into a huge loop to bring that remote control in contact with the gene's starting point. It's an amazing bit of molecular gymnastics, all just to give that gene a major boost.
All right, so you might think once the gene is copied into a message, the hard part is over, but that's not even close to true. Making the decision to copy the gene is maybe half the battle. The control doesn't stop there. Once that little RNA message is made, it has to survive a whole new series of checkpoints before it ever gets turned into a protein.
So that leads to a really fascinating question, right? What if a message has already been sent out from the DNA headquarters? The copy's been made, it's on its way. Is it too late to stop it? Can the cell actually intercept a message in transit?
The answer is a huge, resounding yes. And the way it does it is just incredible. It's a process called RNA interference. Basically, the cell can create these tiny little RNA molecules that act like heat-seeking missiles. They are programmed to hunt down one specific type of messenger RNA floating in the cytoplasm, lock on, and then signal for it to be completely destroyed. Just like that. Message intercepted and eliminated.
And believe it or not, there's even one final checkpoint, right at the finish line, at the protein factory itself. Take the molecule that stores iron. When the cell's iron levels are low, a specific protein comes and just clams right onto the RNA message. It physically blocks the factory from reading the instructions. No more iron storage protein gets made. But when iron is plentiful again, that blocking protein lets go, and the factory is clear to get back to work. It's control right down to the last second.
Okay, let's bring all this home. We've seen simple on and off switches, we've seen complex remote controls, and we've even seen message interceptors. So why does all of this matter? Let's talk about why this incredibly intricate system of control is absolutely critical for our health.
So, the big takeaway here is that gene expression is not just one simple switch. It's this whole cascade of control points. You've got epigenetic control, deciding which parts of the DNA are even open for business. Then you've got transcriptional control, deciding which genes get copied. Then post-transcriptional control, and even translational control, deciding which of those copies get made into proteins. It is all about one thing: making the right protein, in the right amount, at exactly the right time.
And look, this isn't just some abstract biology lesson. This stuff has profound, real-world consequences. Because when these incredibly delicate control systems break down, when a stop signal gets ignored or an on switch gets jammed in the on position, that's when things can go horribly wrong. Uncontrolled cell growth is often the result, and that can lead directly to devastating diseases like cancer.
And that leaves us with a final, really powerful thought to consider. Our cells are absolute masters of this complex language of gene control. So what happens is we get better at speaking their language? What new therapies become possible? Can we learn to go in and correct the typos, rewrite the faulty instructions, and maybe, just maybe, program ourselves for a healthier future? It's a huge question, and the story is really just beginning.